Hello and welcome to the special edition of Meet the Microbiologist. Our guest today is Dr. Roger Lévesque. Dr. Lévesque is this year's winner of CSM Murray Award for Career Achievement. CSM Murray Award is the Canadian Society of Microbiologists' premier award for senior researchers. The award is sponsored by Canadian Science Publishing, the publisher of the Canadian Journal of Microbiology, and we thank them for their continuous support to the Canadian Society of Microbiologists. Dr. Roger Lévesque is a professor at the Department of Microbiology, Infection and Immunology at University of Laval. Dr. Lévesque is also the founding director of the Institute of Integrative Biology and Systems. Dr. Lévesque's laboratory studies bacterial pathogenesis of lung infection in cystic fibrosis. They work a lot on Pseudomonas aeruginosa, one of my favorite organisms, and also looking at the human host response to the infections. Welcome, Dr. Lévesque, and congratulations on your award.
Thank you very much for this introduction. I really appreciate this award. The CSM has been an organization that I've been participating in for a very long time since I started out, so I really appreciate this award. However, I do not necessarily feel senior, so this is going to be an interesting discussion. That's fine.
Let's just establish researcher.
Yeah, I agree.
So great. So Dr. Lévesque, I was wondering to start off if you could share your academic journey with us.
Sure. I'm originally from New Brunswick at Munston. So I grew up in a small town that was a paper mill town, as they say. And very young, I was interested in, of course, my father was a fisherman and a hunter. So I got really interested in nature quite a bit. And then after high school, I was already interested in science in high school. For some reason, I really don't know why. It just came up that science was what I was interested in. I liked the logic and the way things were described very young. So I decided to do an undergraduate degree at the University of Moncton in biology. But the biology course there was quite large. So we had courses in ecology, we had courses in microbiology, and I realized by discussing with different professors that the biology part in ecology, but more so some of those systems involving, for example, nitrogen fixation and so on, was all by microbes. So there was quite an information about the microbes playing different roles in different systems, causing disease in animals and so on. So I really got excited about that. And I decided to maybe go forward and do a master's in microbiology. But before that, I had the opportunity to work with Parks Canada at Prince Edward Island. So I was there for one summer, and then I realized all these different approaches that were used to study the environment, interdital organisms when the waves go up and down. And it was an interesting summer. Then I moved to a larger city, University of Montreal, and I did a master's degree there, working on Staph aureus and Candida albicans and interactions in an animal model and so on. So I got interested in that quite a bit. But very quickly, I realized that I wanted to move forward into another university. So I did a PhD here at Laval University with Dr. Péchet, who was an infectious disease guy working on antimicrobial resistance and looking at enzymes called beta-lactamases, hydrolyzing beta-lactam drugs, penicillin and cephalosporin. So I did my PhD in that. Published some papers, and then decided that I needed to go out. I realized I wanted to be a professor, so I went to Harvard University. And that was quite eye-revealing because at Harvard University, you can have a seminar every couple of hours every day. And it was quite fascinating. The library there is just unbelievable. And I had an opportunity to be with George Jacobi at Mass General. He's an infectious disease guy, but really interested in bacterial genetics and antimicrobial resistance and transposable elements, and that was fascinating. But he told me, Roger, you're gonna probably go back to Quebec, you're French, so maybe you want to go into all these different labs. So across the room, there was a guy working on CMV who at that time was thought to cause HIV, but they wanted to do Southern blots analysis to look at homology of these different viruses. There was somebody upstairs doing the typical dideoxy sequencing starting up. So I had an opportunity to go in about 10 or 15 labs learning all these technologies, physical analysis of DNA and doing agarose gels and looking at plasmids. So I had an opportunity to do all of those different techniques in different labs. And I kept that until today. So after my PhD and my postdoc there, I went to Cold Spring Harbor for a couple of courses in advanced vector genetics. And after 3 years at Harvard and these courses, I came back to Laval University for a position. And I started my lab at that time. So that was in 1984, 85. So, and I built up my lab working on antimicrobial resistance, studying Pseudomonas aeruginosa, looking at antimicrobial resistance, transposons, received an FRS2 award as a young scientist. That award paid my salary for 15 years. So university was quite happy with that. And I got some grants from CIHR and local provincial grants and built up my lab, working on different areas in terms of antibiotic resistance. But I was interested in a lot of different things. So I was looking at different projects, looking at microbial virulence, antimicrobial resistance, eventually got interested in insects and then got interested in genomics. But I realized my lab was at the Faculty of Medicine, not in the hospital facilities. There was a reason for that, because I wanted to do some biology quite a bit. Maybe call it environmental microbiology, I don't know. So I realized they wanted to create this institute. They had this $40 million building coming up. So I decided to move there because there would be people in forestry, people in biochemistry, biology, medicine like myself, and agriculture. And we were sharing some technology and some interest in terms of cell biology, genomics coming up. And that really excited me. So we got together, we were there, but things dragged for a while. Took a long time to sort of organize, and I decided in 2008 that this should be an institute. And I thought we should integrate what we do, have platforms. We have a platform in genomics, we have a platform in microscopy and molecular imaging, and we have a platform in bioinformatics. And I managed to convince university to pay salaries. So we have about 12 people working in these platforms that are professionals. So that really sort of built up the institute. It's a small institute. We're about 27 professors, 300 people with research associate, postdocs, graduate students, but it's well funded. We have 8 Geno Canada projects in different areas. So I think it's fascinating to be here. And that sort of got me going until now.
Wow. This is really interesting. And I think the lesson for all of us, especially students who are looking at microbiology as career, environmental microbiology and nitrogen fixation was the hook that pulled you into microbiology.
It did.
And here you are.
Yes.
It's fascinating, and it's really interesting to actually learn about our journey through our career, how it starts and where we end up. So keeping open mind, and I think, as you said, exploring different options is so important to find your love, right? So that's quite amazing. And Dr. Lévesque, your work on Pseudomonas pathogenesis, particularly, at least from my own interest, is the regulation sigma factors in Pseudomonas, how they play a role in virulence and pathogenesis is fascinating. So tell us a little bit about what's going on. I know there are multiple things going on in your laboratory, but what's the focus now?
Well, one aspect I didn't mention is, as I was building my career, one thing that really made my career was the Canadian Bacterial Disease Network that was developed by Bob Hancock at UBC. And I had the opportunity to work with people across Canada on different aspects of infectious disease and different ways to look at the cause of infectious disease, microbial virulence, using classic microbiology, using what we call today proteomics and bacterial genetics and genomics coming up. So that really helped me sort of open my eyes what's going on across Canada with all these different labs at UBC, Toronto, McMaster, McGill, Eastern Canada at UNB, and so on. So we had an opportunity to have all these different researchers across Canada being part of this group. And we would have an annual meeting and we'd have projects that were well funded. And that really made me realize the extensive aspects of microbiology. And I started to focus more so on Pseudomonas aeruginosa for a number of reasons. I had already worked on transposable elements as a postdoc, but I wanted to look at microbial virulence. But the strategy for me was, at the time, Pseudomonas aeruginosa genome, PO1, was not known. And I tried to look at different ways of this, and that's when I developed signature tag mutagenesis. The idea was to use my expertise on transposable elements, but build transposed elements with markers that we could use. PCR was coming up at the time. So we wanted to put tags on plasmids and then make libraries of PO1 and then sort of screen that in the rat lung model of infection to see if we could isolate mutants that would be defective in vivo maintenance. So that was the spot on and we started with. So we started playing with this, and we published several papers on this with the technology, but also we realized by doing this that we could pinpoint genes that if they were inactivated, the Pseudomonas aeruginosa could not maintain itself in the rat lung after 6 or 7 days. So we sort of pinpointed those. And we used Sanger sequencing to sort of find the endpoints to see which genes were involved. And to make a story short, we realized that 45% of the genes were in unknown open reading frames. So there was a lot of genes or products of those genes that play a role in vivo maintenance that were not known. Of course, the system was elegant because we did pick up genes, for example, that were inactivated, but were classically known as important in virulence, LPS production, production of biofilm, production of capability to pick up iron from the host and so on. So it was an elegant way to look at it. So I pushed that very hard to look at a whole series of genes. And then I developed ways to look at this and sort of refine the rat model and the mouse model to look at animal modeling and sort of detail out these genes. But as that was going on, there was a tremendous explosion in bacterial genomics, of course. All these papers were coming out, sequencing the first Haemophilus influenzae, published by Craig Venter and then his team with Hamilton Smith. And then there was all these papers coming up. So I really got excited by that. And I got involved in doing microbial genomics and trying to look at different strains of Pseudomonas aeruginosa from the environment, from different infections in the host, from different human infections, but also animal. And with Cystic Fibrosis Canada, I built this database of the International Pseudomonas Consortium database. We have over 2,000 strains and some metadata that's on a website that anyone can request strains. So I've been giving thousands of strains around the world to anyone. And the contribution was from pseudoscientists around the world that wanted to have that collection built up. And the requirement is that I would give out the strains to anyone that would like to have it to do some research. And of course, the agreement has to come from the original provider, not me. And that has been a fantastic database. And it's complementary to pseudomonas.com by Fiona Brinkman, where there's a lot of information about the genomics. So that's been an exciting part of the work that I've been doing. So, and I guess the CSM award that I received is pretty much around what I've done with Pseudomonas aeruginosa. Then I pushed into other fields. I've been looking at LPS, cell wall biosynthesis, use phage display to sort of isolate inhibitors of cell wall biosynthesis. And that was picked up by certain companies that wanted to have these, because you had to produce the MUR-H and MUR-A proteins to do that. And because we could do that with Pseudomonas aeruginosa, we had MUR-A, MUR-B, MUR-C, MUR-D, MUR-F produced. We could purify the protein to use in assays. And some pharmaceuticals have been using that, and universities and collaborators in the UK. So that was a fun part. And also bacterial cell division with phage display. I developed inhibitors for that. And we're doing a bunch of different things now. So now we're moving very recently to 3D bioprinting using human lung cells. There's about 50 of them, but we're using now, just starting up with 3 or 4 cell lines to try to build from scratch some sort of primitive lung system that could be used to study bacteria host interaction with Pseudomonas aeruginosa. And I think that's pretty exciting.
Really fascinating. And one of the things I think Dr. Lévesque, thing that I really appreciate coming out of your lab is, as you mentioned, sharing of these resources. Best science comes from sharing, and the fact that you have these amazing resources and you're openly sharing those with researchers around the world. And I think that sort of contribution goes a long, long way towards advancement of the field. So that is fascinating, and I think that's an important part of what we do as scientists and researchers. So my next question is actually sort of a 2-part question, as a established researcher award winner, when you get someone knocking on your lab door, seeking a position, a graduate position, for example, in your lab, what do you tell them if somebody comes in asking, well, I'm considering a graduate position? So that's part one. And the part two is basically, what do you tell a new investigator, a PI, a new assistant professor coming into your institute? So
yes, okay, that's a very good question. I like that. Well, I mean, the first time that I meet graduate students, I mean, they come to your lab, but they want to be a graduate student. And they say, well, I want to be a graduate student, I want to be a PhD, and then I want to be a professor. I go, hold on there, whoa. Let's start with the beginning. Why do you want to go into a lab? Why are you interested in doing a graduate degree in microbiology? What's your feeling about that? Why? And of course, undergraduate students move into the graduate's first year. They don't really know, but they're interested. So it's just trying to pick their brain to understand where the interest comes from. Is it from the undergraduate courses? Is it from the motivation of certain professors that gave these undergraduate courses? And then you try to define, what do you want to do? What's your interest in microbiology? It might not be in my lab. And I would not certainly pick someone in my lab that has some interest in something which is not in my lab. But I would certainly give references to key labs that I know would be very good, so that that person can go to according to his interest. That's really critical. You have to be curious and interested in, but of course it's so large in microbiology and I do many different things. So I usually find something that the student is interested in. The first thing I also do is I make them visit the lab, and then I sort of discuss the projects a bit that we do. And then I tell them to sort of, or her, come back and talk to my people in the lab. And I'm not going to be around. So if I say, look, you want to come this afternoon, tomorrow morning, whatever, come in the lab, I'm not going to be there. You talk to people in the lab and discuss what's your interest, what you want to do, why do you want to do this, and where do you want to go if you want to do a master's job, you're looking for a job. I have students coming, oh, I need a master's and I want to work as a research associate, or I want to work company. Fine, that's not a problem, because not everyone can be a professor. Because you have to go for the PhD. Today it's 3 or 4 years of postdoc. It's very competitive, grant writing. One thing I do that I enjoy is I say, okay, so if they pass the first steps, I find the interest, they want to come, I give them a 1-page and I say, write to me, what is microbiology for you? What's your interest in microbiology? I mean, you think about it and then you hear about it, but when you write about it, it's very different.
Right.
So that sort of crystallizes what they want to do. And then I add, if there's a project that's of particular interest, maybe just write a few lines about that. It does two things. I also make them write in English. It does a couple of things. It makes them realize what they want to do, but also it shows where they are in the English language, writing and speaking, because I do talk to them in English. There's no way you can do science today without being good in English. Some people are very good, but they're not interested in going higher up because of the writing that's necessary and critical to go after the PhD for a postdoc and get a professor position. It's very competitive, but some do. And I try to stay open. I give them, and also I try to make them read as much as I can. I do read a lot of different things. And I try, in my group, I sort of send every Thursday or Friday papers that have just came out in Nature, Microbiology, whatever. It might be on 3D bioprinting, it might be on CRISPR-Cas, it might be on stuff that I'm interested in, rapid technologies to diagnose. I think one way we have to realize is that microbiology today is technology driven. I'm not saying we should all be in technology in that, no, but if you have some interesting microbiology questions, you have to look at the tools. And I've been lucky, I have some good funding, so I've got the tools to do a lot of different things that I want to do, but it's not the case in every lab, unfortunately. But the tools are critical. I mean, I have a picture of Pasteur at the back of me, and I've been at Institut Pasteur because I have a weakness for him and for what they've done, what they do today. But things have changed from a Petri dish and sort of putting in bacteria. Things have evolved dramatically. We still need that. We still need to identify bugs, and we still need to do some phenotypic analysis, but you have to realize the palette, like a painter, of what you need to do all these different things in a project. And I think it's just fascinating. I'm really excited, probably much more than when I started out, because now I have the experience to do all kinds of things that are fascinating. So, yeah.
This is actually, it is very exciting. And I think, one of the things that we have seen in the last 2 years or so, going through these unfortunate events of pandemic, has also sort of shown us the power of science and technology. What we have been able to sort of achieve in last months would have been inconceivable to achieve in decades, few years ago. So it is, with every challenge comes an opportunity, and I think this is what we see, or at least from microbiology side of things. So moving on to the other part of the question, for a new assistant professor coming to your institute. What sort of advice do you give them? I mean, you did say that writing grants. I mean, that's a lot of work these days, and it can be fairly frustrating at times. So,
yes, I think the key element, if one is looking for a professor position, is to be ready for that. That means to have a solid postdoctoral training. I've seen people go into different universities and departments with only 1 or 2 years of training as a postdoc, and then they're on their own. And I think that's really unfortunate. That should not happen today, because the competition, but also the complexity of the research process and getting grants is quite something. I'll give you a couple of really realistic examples. For example, Larry Goodridge at University of Guelph, with whom I work closely as a young professor. So I've been a mentor for him for quite a bit. We got some large grants from Genome Canada. He's an expert in food microbiology. So we got a grant on which we call cystomics of Salmonella, which was a very large grant. We got that. He was the leader, and I was the co-leader, but he was a young professor. So that was a concern for Genome Canada, Genome Quebec. But I have the experience, so we sort of work as a team very closely. And we did that for 4 years. It just finished this spring, and it was a fantastic project. We developed this large collection of Salmonella for people around the world, more than 5,000 isolates from different food, human infection, whatever. We published over 50 papers on that part of the work. And we trained research associate, postdoctoral fellows, graduate students, technicians, and so on. So Larry is quite excited. Then we got another grant starting this last year, which is called Sentinel. It's monitoring enteropathogens, but in wastewater. So we're doing that. And of course, we were lucky to have that funding, because not only enteropathogens, but also, which include 6 bacterial species, 3 viruses, and 3 parasites. So we're monitoring that in wastewater every week. And I've been doing that in Quebec, the Quebec wastewater system. And of course, the COVID came up. So we really pushed very hard to detect COVID in wastewater. But one has to realize that the amount of water going through to get sort of purified, cleaned up, is 8 to 10,000 cubic meters of water per hour.
Right.
So how are you going to detect a COVID, an RNA virus in that? So we developed the techniques with other people across Canada. There's now an organization across Canada trying to do that in every province and many cities. We managed to do that. But we also developed the technology to see if we could use metagenomics, genome assemblies, to not just use the 16S that people sort of use. This is a Salmonella, but that's it. Or this is like maybe a Listeria or a Vibrio, but is it Vibrio fulminicus or what is it? But with long-read sequencing with Oxford Nanopore, we're able not only to detect these microbial species, but we can assemble most of their genomes and find out, for example, E. coli, does it have this Shiga toxin or not, and so on. So we can do that, even if we know how much E. coli there are in wastewaters, fecal and other pathogens and so on. So we can do that now. This is something that the Public Health Agency of Canada, collaborating with us, is going to use. So we're pushing this very hard. And the COVID, we've been sort of following the COVID since June 2020, because we were locked down for a bit. But our data correlates very well with the cases that are in hospitals. So there's a nice correlation. And now the Quebec government is really interested in using that. not only to monitor what's going on in the pandemic without using PCR, but maybe in the midterm, we could sort of predict or see if we can sort of see coming other pandemics. It could be influenza, it could be something else. So there's a keen interest for the future to use these technologies and apply them in the environment, but also in the infectious disease.
Very exciting stuff.
Thank you.
I would like to thank you again for your time, Dr. Lévesque, and many congratulations. And I really look forward to seeing you at the Canadian Society of Microbiologists meeting this year.
Yes.
So far, everything looks good to be in person. So hopefully, it's been 2 years since we gathered. So really looking forward to this gathering and looking forward to your talk as well. So thank you again. Thank you for your time and congratulations.
Yeah, thank you very much. I'm looking forward to seeing Larry Goodwin at Guelph and then Cesar Crisiguera and other collaborators. So I've collaborated with many people around the world, but these people are really key to me and very important. Thank you very much.
Thank you.